Okay. Today we're going to be doing a little bit of a review of DNA in this lesson. Please feel free to pause the lecture at any time so that you may keep up and write these notes down. In order to receive full credit for your notes, you need to have all of the information written down as well as extra things that I have you add to your notes. So please listen carefully. If you recall, we had this thing called Chargaff's rules, and Chargaff noticed that there were equal amounts of adenine and thymine, which are the nitrogenous bases that are found in DNA. So adenine must pair with thiamine, and our guanine must pair with cytosine. And we abbreviate those as A, T, G, and C. I always remember that adenine and thymine go together because they are made up of straight lines. The A's and the T's are. And then the G's and the C's both curve. So you see the curve in the G and the curve in the C. So they pair together in DNA. The bases, as they connect together in DNA, form weak bonds that are called hydrogen bonds. And those are important because they need to be strong enough in order to hold the nitrogen bases together, but weak enough that they may be broken apart when we replicate DNA in our cells to make more cells. Here's an example of A and T pairing together and G and C pairing together. Notice on the A and the G, you see two different rings, and the C and the T are just singular. And we'll talk a little bit more about how we can classify those. Another thing to notice is on the G and the C, there are 3 hydrogen bonds represented by these dotted lines. And here, between the A and the T, there are only 2. You may have noticed that when we put our magnetic models together in class, that some had 3 magnets and some only had 2. If you recall, when we watched Photo 51, there was a lady by the name of Rosalind Franklin, who was a scientist who did X-ray crystallography to take pictures of the DNA crystals and was able to develop them very well. We had Watson and Crick, who used the information that they obtained, I guess illegally, from Rosalind Franklin to build the first DNA model, and they were awarded then the Nobel Prize for the discovery of DNA. That is Rosalind Franklin. And if you look here at this picture, that was the famous Photo 51. This X-shaped pattern let people know that the DNA was in fact in a helical or like a twisted structure. We'll go a little bit more into the structure of DNA now. It has the 2 strands that are coiled or twisted. We refer to it as a double helix. Because of the 2 strands that are on the outside, it's double, and because of its shape, it's helical in structure. We call the backbone of DNA, or if you're thinking of it as a ladder, would be the handles or the outsides of DNA, are made up of a sugar called deoxyribose that is bonded to phosphate. So you have these sugars and phosphates that are bonded to each other that repeat over and over again. The sugar and the phosphate are connected by what are called phosphodiester bonds. And then in the middle, which would be like the rungs of a ladder, are made up of those nitrogen bases. And those are what are connected by those hydrogen bonds. Okay. So on the left-hand side of your screen, there's just showing how the DNA is actually helical in structure, and you're familiar with that from looking at the models and from other things that we've talked about. On the right-hand side, it's kind of more of another simplification of DNA, showing it as a ladder. and you would have the legs of the ladder being the phosphate and sugar backbone. And then the rungs of the ladder would be these nitrogenous bases, these A's, T's, G's, and C's. And please put this diagram of the ladder into your notes. And you may pause it if you need to. DNA itself stands for deoxyribonucleic acid. And so you have your DNA. And we'll talk about what RNA stands for in the next lecture. It is made up of those nucleotides. And remember when we talked about nucleic acids being the fourth macromolecule? DNA is a nucleic acid. Nucleic acids are made of these nucleotides. And a nucleotide consists of the phosphate and the sugar, which we see on the outside of DNA, as well as the nitrogenous base. So with the structure of DNA, you have two nucleotides that are paired together. And they're connected once again by those hydrogen bonds. Okay. So if you're looking here, we have our phosphate group paired up with our 5-carbon sugar and our nitrogenous base, which is just a generic nitrogenous base, A, T, C, or G. Okay, and notice here on the sugar you have carbon 1, and that's what the C stands for. So carbon 1, carbon 2, carbon 3, carbon 4, and our 5th carbon is here. And this is going to be important to understanding the structure and the arrangement of the different strands of DNA. So you have your phosphate group, and it's called PO4. So P, your 4 O's, is PO4. Okay, so looking a little bit closer at the sugar, carbons are usually numbered clockwise, and they're numbered 1 through 5 on here. So here's your 1, 2, 3, 4, and 5. Please sketch this into your notes as well. Okay, so here is our 2 strands of DNA connecting in the middle. Oops, excuse me. So here you have your sugar, phosphate, sugar, phosphate, sugar, phosphate, and you have the same thing going on this side. Here would be our guanine paired with our cytosine in the middle, and our thymine paired with our adenine. Okay? These dotted lines represent the hydrogen bases, and you can notice on the sugars they've been numbered 1, 2, 3, 4, and 5 for our carbons. And here we have 1, 2, 3, 4, and 5. Comparing the left side of sugars and phosphates to the right side of the sugars and phosphates, you may notice that they are flipped upside down. So you have, in this case, the 3rd carbon being here and the 5th carbon being here. And over here we're looking, your 3rd carbon is now here and your 5th carbon is here. Okay. This is because our DNA strands run in an anti-parallel fashion, which one strand goes from this 5' end to the 3' end. And that is looking, I'll go back real quick, that is looking at these sugars. So you have one that goes from this 5' to 3', and here you have this one running 3' to 5'. When we talk about DNA replication in the next lesson, it's going to be important to know the difference between the 5' and the 3' strands of DNA. Okay, so one strand goes from 5' to 3', and the other strand goes in the other direction. This is a simplified model of DNA unwinding so that it may be replicated. So you have your 5' to 3', your 3' to 5'. Okay. You may have noticed that on the different nitrogen bases, some had double rings and some had single rings. The double ringed structures are called purines, and our adenine and our guanine are purines, and our single rings are pyridamines, and that is thiamine and cytosine. Okay. You'll have one purine that pairs with a pyridamine. You cannot have two purines that bond together, and you may not have two pyridamines that bond together. That's why you would notice when the nitrogen bases were held together, guanine and cytosine had 3 hydrogen bonds. And so you see your purines and your pyridamine. And you have these 3 hydrogen bonds. The A and the T are only going to have the 2 hydrogen bonds. Okay. So once again, you have your pyridamine and your purine, and they have these 2 hydrogen bonds. This is a question that I'd like you to answer in your notes, and this should be the last thing that you'll add to this section. So if there's 30% adenine, how much cytosine is present? Please use what you know about base pair rules to answer this question into your notes. When you are completed with all of the notes, you may submit them to Mrs. Campbell to be checked in order to move on to your next activity. Thank you.